Hello everyone and welcome back to the session 2 of week 8 of NPTEL course in Herbal Drug Technology and Regulatory Sciences. In the first session, we saw important books called as pharmacopoeias and we saw herbal monographs. So whenever we want to do quality control of any herbal materials, we see that this monograph act as guidelines, and depending upon the pharmacopoeias we have different type of monographs. In this session we will go little deep into this quality control parameters which were used in the monograph. So this quality control parameters depend and are different from countries to countries. However, considering the recent surge in the global trade, WHO laid its own guidelines for quality control of herbal drug materials. Now what happens is developed countries have a very stringent guidelines and developing countries may have those quality of materials, but just because they couldn't carry out the quality control methods in that or couldn't spend that much quantum of their finances and resources on quality control, that makes their raw material unacceptable. So WHO kind of lays down a minimal guidelines, a threshold which all the drug materials should meet for global trade as well as domestic trade to ensure three important parameters. So WHO lays down the guidelines to assess the evaluation of drug based on quality, safety and efficacy. Quality will tell you how good the drug is, safety will tell you how safe it is and how free it is from pesticides, radioactive, aflatoxins and other bacteria, and efficacy will tell you are the medicines really efficacious. So these three criteria are the basic criteria which needs to be met by most of the herbal materials. Now in addition to that, WHO also ensures or has laid down the guidelines to ensure that this guidelines or this quality control parameters act as a good framework for national regulatory authorities. So you can see the same parameters which we will be studying today are a part of many monographs. It can help scientific organization in standardization of their drug material as well as manufacturers to frame their own guidelines if they want to import, export or use the herbal drug materials. This is generally done for assessment of quality, documentations and submissions once the product is prepared. So if you have to see what WHO guideline is all about, let's see the little contents of it. So these are the general contents of WHO guidelines. Let me show you the WHO quality control guideline booklet and then we'll take a quick run through all this quality control parameters. So here's the booklet of WHO guidelines. So if you see quality control methods for WHO guidelines, this is a small booklet just about 122 pages and it describes the quality control parameters much in depth. So I'll just give you a brief pretext to this. So this explains the criteria, the guidelines, why it is prepared, publication data, the contents which we just saw. The contents includes something like general notices. This is generally giving about temperature, units, how it should be stored, process, powder fineness and sieve size, how sampling should be done, and later on different quality control parameters such as ash value, extractive value and so on. So let's just see few of them here. So if you want to check the solubility of a herbal drug material, so if you want to see the solubility of herbal drug material, what you need to do is just to take a gram of herbal drug material and try to dissolve it in a solvent. Suppose if this 1 gram is dissolving in 1 part, that is 1 ml of solvent, it's said to be very soluble. And as the ratio increases 1 part of drug to 10 parts of solvent, we call it as freely soluble. About 10 to 30 we call it as soluble, and if the drug requires more than 10 liters, that is more than 10,000 parts of solvent, we call it as practically insoluble. Similarly, another criteria which WHO lays is about the particle size. So, when you have herbal powders, they come in various sizes. We call it as mesh sizes. So, in pharmaceutical chemistry, when you say that this is a particular mesh size, the mesh size of WHO and the mesh size of Indian pharmacopoeia varies. So, WHO mesh size is much easier. Let me tell you how. So, you can say if you have to describe the powder as coarse, it should be 2000 mesh size or sieve size, moderately coarse between 250 to 710, fine 180, very fine 125. Now, WHO mesh size are very simple. If I say WHO mesh size is 2000, that means the particles are 2000 microns in size or less. So, you can see a table here. So, it is like a nominal size of aperture and diameter of wire, but what is important to us is if a mesh size is 2000, this includes micrometer. So, anything which is like a 2000 micrometer will enter in. So, 7110 indicates that a particle size of 710 is there. So, 710 micron particles will all pass through. So, here WHO has an advantage in sizing that the mesh size is same as the particle size of the powder which passes through in micrometer range. Now, moving on to other criteria, let us get back. So, other quality control methods, you can divide it broadly into organoleptic, microscopic, macroscopic, physical, chemical and biological. So, organoleptic, when I say it's something which I can sense by organs, macroscopy is something which is again a organoleptic parameter which can be seen or perceived. Then microscopic is a parameter which you observe under microscope, right from your simple microscope to compound microscope right to SEMs. Physical criteria includes the physical properties. Chemical criteria include the chemical evaluations including the phytochemical evaluations, and biological criteria include the microbial estimation, even the efficacy studies. So, let us study few of this criteria starting with organoleptic. Now, when I do organoleptic evaluation, I kind of visibly see the color. You will see that some leaves of some drugs are lighter, some leaves are darker. You can see the color difference between what are called as nutgalls, which may vary from grey to green. It is said that the nutgalls are generally grey when the insect escapes it and green when they are harvested in the proper right time. So, the timing and the tannin content can be estimated just by knowing the color of the galls. So, that in that case the color plays a very important role there. Not only that, odor is a good characteristic of essential oils. So, spices and condiments which are carrying essential oil, if you get a very strong aroma to it, that means the essential oil is intact in them. But if you are not getting aroma out of it, that means somewhere your drug which is brought into your industry is exhausted. Taste, there are drugs which are bitter, there are drugs which are extensively pungent, astringent. So, bitters, you have something like your cinchona, you have something like your neem, kalmeg, that is your andrographis, swertia are all examples of bitter drugs. You have pungent such as your chilies, black pepper for their alkaloidal content. Then you have tannin containing drugs which are extensively astringent such as your tea, amla, and then there are sweeteners, especially natural sweeteners such as stevia, whose leaves the moment you put in mouth you will get a little sweet lingering taste. Not only that, you can also analyze texture just by pressing the leaves within the hand. You can come to know which species it's belong to. I can give you a simple example. If you are dealing with two species of senna, that is your Cassia angustifolia and Cassia acutifolia. In that case, you see your Cassia acutifolia, that is your Alexandrian senna, is much thinner then Cassia angustifolia, which is your Indian senna. So that consistency, even for drugs which contain good amount of mucilages, after you pour water on them, if it's giving mucilaginous consistency, I can say it is genuine. But if the mucilage or the slimy layer is not formed, I can say it is a adulterated or some other substituted drug. So you can check for that. Even in cases of gums, you can actually feel the swelling between your hands and fingers and see the texture or analyze the texture of it. Now moving to the macroscopic analysis, here you can see different parameters which are very typical of the drugs. Say for example, when you have your dalchini or cinnamon, cinnamon is known for its typical quilled structure. As you can see here, this is what is called as quill, a typical C shape, which may further be rounded more to get what are called as more quilled, or sometimes one quill is inserted in the other to get what is called as compound quill. At the same time, for cinnamon, you will see that if you are talking about Cinnamon zeylanicum, you can also see that the outer cork layer is missing in that. At the same time, when you are seeing quills, you can see another drug called as kurchi. In that case, the quills do not occur like this, they occur in the opposite side, and that's where you call it as a recurved quills. So the outer bark folds in extensively outwards so that the inner part is completely exposed. You can also check for tiny appearances of organelles such as lenticels or presence of any infections or disease conditions in the plant. So macroscopic evaluation is very important. Now moving on to microscopic evaluation. Now WHO lays down sample preparation, reagents, how it should be prepared, how it should be mounted. All the detailed guidelines about microscopic evaluation are given in. So say for example, if you have leaves, so for thinner leaves you might want to clear them in chloral hydrate. So how the specimen should be picked, how it should be teared, how should it be cleared, how much chloral hydrate to be used, when it should be used, when it should not be used, what are the other staining reagents. So whenever you have water, can you mount it in water, or is lactophenol more better? So even with staining, so all the preparations, all the reagents, staining reagents, bleaching reagents, mountants, the WHO gives a good detail of all of this. It gives you also some studies based on monographs about transverse sections, epidermal leaf preparations, as well as identifications. So using this guidelines, you can prepare your sample, place it under microscope, use a suitable mounting agent, and then you can observe. Say for example, very characteristic parameter of Vasaka leaf is a diacitic stomata. So you can check for diacitic stomata, you can check for specific identifying features such as calcium oxalate, especially like you see here, the rosette shaped ones, the rosette calcium oxalates of Arjuna bark. You can also see a good transverse sections. In transverse section, you can see the whole botanical arrangement, which will help me in identifying the plant part. If I stain it properly, I can even do what is called as histochemical localization. Not only that, it also gives me identification. So I can identify my plant parts, I can take care of it. All of that can be done based on WHO guidelines, for which you have a good protocol or SOP for slide preparations. It also gives us an idea about leaf constants. So leaf constants are parameters which are constant for a particular plant species. This includes parameters like stomatal number. Simply, it is the number of stomata per millimeter square. So if you take a leaf, and you focus in the area in between the leaf, that is between the midrib and the edges, and then you count the stomata per millimeter square, what will happen is you come to know how much stomata it contains per millimeter square, and that is called as stomatal number. But keep in mind, the stomatal number differs on upper and lower epidermis. Always in many cases you will find the stomatal number is higher on the lower epidermis as compared to higher epidermis. Similarly, we have what is called as vein islet and vein termination number, which is corresponding number of vein islets or corresponding number of vein terminations within a 1 millimeter square area. Simultaneously, you also have what is called as stomatal number, which is actually a ratio of number of stomata to number of epidermal cells counting stomata. So, stomatal number is given by a formula S divided by E plus S, where E is actually epidermal leaves and S is stomata. So, S divided by E plus S into 100, that is expressed as percent, is called as stomatal number. Another leaf constant is your palisade ratio. So, you can see here epidermal cells. So, beneath each of these epidermal cells are located many palisade cells. So, number of palisade cells beneath each epidermal cells is called as palisade ratio. So, you basically count the palisade cells and then divided by number of epidermal cells, that ratio is called as a palisade ratio. These are all termed as leaf constants, and WHO takes a note of them as a good quality control parameters. Moving on to the next one, and that is your physical methods of evaluation. Your physical methods of evaluation includes determination of foreign organic matter, that is the extra contaminants that come in your product, moisture content, extractive values, ash values, foaming index and swelling index. Now, coming to foreign organic matter, as the name indicates, foreign organic matter, that means something which is not belonging to the drug, that is foreign to the drug. That means any extraneous matter of the plant or non-plant material which is present and should not be present ideally, or in many cases it should not, or it is not derived from the same source, and as a result it's come in as a contaminant in your sample. All of this are called as your foreign organic matter. So, WHO describes foreign organic matter as plant material or materials plural other than specified within the limits of the plant. Say for example, if clove is a clove bird, and if you see a stone cell in that, that stone cell does not come from bird, that stone cell actually comes from stem. So, for that definitely a foreign organic matter is there. Then another living organisms, ants, grasshoppers, lizards or pieces or parts of them, and even any mineral or any other extraneous matter such as your soil, stone, sand, dust. So, what is done is generally for determining foreign organic matter, a complete or unground drug is preferred. In that case, it makes it very easy to identify, because once you pulverize the drug, even the foreign organic matter gets pulverized and it becomes difficult to separate visibly. So, generally for visible separation, the whole material, the sample size depends on the material. Say for example, if you are dealing with roots and rhizomes or barks, you can take half kg of it. If you are dealing with leaves, flowers, seeds which are little lighter, you can deal with, like, you can take a sample size of about 250 gram. And if you have cut materials which are average fragment is less than half a centimeter or so, then even in that case you can just take 50 gram of sample. So, take this sample, spread it on a paper and start observing with the help of magnifying lens with about 6x or 10x magnification, and start picking up those foreign particles. In many cases, this drug material is sifted through a 250 mesh, and what is done is whatever is coming off is all treated as dust, which is a mixture of ground plant material plus your extraneous matter. So, that is also treated as foreign organic matter. So, you will have dust being removed, then you take your sample and still identify if you can have more of this foreign organic matter being there. So, whatever is there, you will take it, weigh it, and if it is half kg, that is 500 gram. 500 gram gives me this much material, so I express it as percentage and I get what is called as percentage foreign organic matter. Now, moving on to the next test is moisture content. Now, moisture is integral part of most of the herbal drugs because they are plant in origin. They have been having water in them till the stage they were processed and harvested. So, once the harvesting is done, it is dried to ensure that the enzymatic processes stop. At the same time, when moisture is removed, the bacterial contamination also decreases. So that is a good way to store the plant, and in some cases such as digitalis, you will see that if the moisture content remains as it is, it will eventually deteriorate your cardiac glycosides which are present in digitalis. So for such determinations, WHO gives you two methods. One, what is called as azeotropic distillation, and second, what is called as loss on drying. In the first method, that is azeotropic distillation method, it uses this special assembly. It's also called as modified Dean-Stark assembly. So what is done here is your drug material, generally 2 to 4 grams or whatever is the specified quantity, is taken and it's distilled in presence of toluene. Now toluene is a little lighter solvent. So toluene being a lighter solvent, what will happen? Your toluene as well as the moisture in your drug will distill off. It will rise up because it's still heated, and then it gets condensed via condenser. Now this time it won't revert back, but it will fall in the collection assembly. Now as it is falling in the collection assembly, it is condensed. So now it separates in two layers. So water will be the denser or the lower layer, and toluene will be the lighter layer. So what happens eventually? The toluene, which is a lighter layer, as more and more distillation happens, this toluene is recycled back. Now this process is continued for a period of 2 hours, and then you see the reading. So you can see there are graduations out here. So you can see the ml of water collected. So this ml of water is present in say 4 gram of drug. So you can convert it into percentage, and that's what gives you percentage moisture content. Now this apparatus can be previously calibrated by deliberately adding 2 ml of water, and then after 2 hours checking if you are getting those 2 ml back. So if it is perfectly right, that is the time you should be keeping for distillation. A more simpler method but slightly inaccurate is loss on drying. Now why I tell you inaccurate is because loss on drying is non-specific. In this case you will get your water being distilled out, but loss on drying also evaporates the solvents present in the sample and also evaporates the essential oil. Say for example, if your sample is clove, along with moisture your clove essential oil will also deteriorate, and hence it's slightly little more inaccurate. So this is done in a wide bottle referred to as LOD or loss on drying bottle. You keep about 5 gram of sample exposed to a temperature of 105 to 110 degrees Celsius for a period of 3 to 4 hours. Then you cool it in a desiccator with the lid closed for about 30 minutes, and then you weigh. Heat it again, you just check if the more losses are happening. If I have reached a good constant weight, I say that much is the moisture content. So say for example, I started with 5 gram, but after 4 hours it is 4.5 gram, that is half gram is lost. That means this half gram per 5 gram is treated as the moisture content. So convert it into percentage, you will get the moisture content percentage. Now moving to the next parameter. The next parameter is called as extractives. Now extractives are very important because they give you the amount of substances that are dissolved. So if we go by principle that like dissolves like, if I am extracting my drug with water, all my polar compounds will dissolve. If I am extracting my drug with something like alcohol, I will have my mid polar compounds being dissolved. If I am extracting my drug with something like ether, I will have my oleaginous or lipophilic compounds being dissolved. So this is generally done to check the compounds present in a sample. So in this case, WHO says you take about 4 gram of sample, and then what you can do is you can extract it by 2 methods. So you can extract it by hot method or you can extract it by cold method. So in hot method, you take this 4 gram of sample, boil it in 100 ml of solvent, and then after so what you do in hot method is you take 4 gram of sample, you add 100 ml of solvent, you kind of swirl it and allow it to imbibe the solvent for 1 hour and subsequently reflux it for 1 hour. So you require 2 hours time, one that is it's kept cold and second that is kept reflux. Once you reflux it, then you can filter it, evaporate about 25 ml of it and determine how much residue is there. If 25 ml contains this much residue, how much ml or how much residue will be there in 100 ml or equivalent to 4 gram of drug can easily be calculated. Similarly, cold method is used for drugs which are not temperature stable. In that case, what is done is such drugs are swirled at room temperature for about 6 hours and then they are kept steady, unshaken for 18 hours. So, after 24 hours, then they are filtered. Same, you take about 25 ml of the filtrate, evaporate it to get the residue, and you can say that if 25 ml is giving me this residue, how much total residue is present in 4 gram, and then I can convert it to the percentage. So, you get what is called as alcohol soluble if the solvent is alcohol, ether soluble if the solvent is your ether. So, this is normally done by heating it in the water bath till the solvents evaporate. For water, you can even use your oven at 110 degrees Celsius. So, good example of that is if you check for amla, the ethanol soluble extractive is not less than 30%. So, that extract, so if I am taking, what does it mean? If I am taking 100 gram of amla, 30 gram should be soluble in alcohol, or if I am taking amla, at least 40 gram should be soluble in water. So, that is what the extractive value is all about. Moving on to our next parameters, they are your ash value or often referred to as residue on ignition. Now, WHO says you can take about 2 to 4 gram of this material and incinerate it. You can incinerate it in a quartz, silica or platinum crucible, completely heat it till you reach a temperature of about 500 to 600 degrees Celsius for an hour's time, and then you can check once the ashing is done how much ash is produced from 4 gram of sample. Then you calculate in percentage, you get what is called as total ash. Now, this ash is actually chemically what is called as minerals and metals. So, your carbon or organic content all goes as oxide like carbon dioxide, nitrogen oxide, sulfur dioxide and so on. So, what is left is inorganic and mineral metal contents. So, this ash is an indicative of that. So, total ash gives you the total content, whereas if you want to specifically check for adulterants like soil, these are referred to as non-physiological minerals because they come from outside. So, what you can do in that case is you can do what is called as acid insoluble ash. So, you can take that ash which has been prepared, boil it in about 25 ml of 2 molar hydrochloric acid, filter it and then wrap that filter paper, incinerate it one more time. This time whatever is physiological like calcium, magnesium will get converted into subsequent chlorides when they react with HCl. So, calcium will form calcium chloride with HCl, which is water soluble. Magnesium will form magnesium chloride, which is again water soluble. Sodium will form sodium chloride, which is water soluble. So, most of the physiological ashes become soluble. What are left on filter paper are non-physiological ashes. So, acid insoluble ash is a good indicator of what are called as non-physiological minerals or materials. Similarly, you have water soluble ash which will give you more exclusively of your physiological minerals. This is done by treating or boiling the ash in 25 ml of water, filtering it and then evaporating the filtrate, that is the 25 ml water which we use, which will give you a physiological ash. Then for drugs which are very sticky or resistant to ashing, you add about 1 ml of sulfuric acid during the ashing process, and what you get is what is called as sulfated ash. So, again giving you an example for amla, you can see total ash is not more than 5%. If it exceeds 5, that means some extraneous soil, dirt or mud is being added, and acid insoluble, that is what is called as the non-physiological ash, should not exceed 2%. Now, going to the next values, this is to do with the determination of compounds which are called as saponins, and this value is your foaming index. Now, we know that saponins by their nature have the ability to form foam. A good example of saponin containing drug is your amla, rita, shikakai. So, especially if you see a shikakai, it gives you a stable persistent foam. So saponins are determined by what is called as foaming index. So what you need to do is you just take 1 gram of your drug, boil it in 100 ml of solvent, reflux it for half an hour. So what is going to happen? All the saponins will dissolve it in water. Filter it, your filtrate will have all your saponins. Then you create this series of tubes. Now what these tubes contain is, now we have had 1 gram of sample. Now this 1 gram of sample is extracted in 100 ml. In the first tube, we will add 1 ml of my extract and 9 ml of water. Second tube, we will add 2 ml of extract and 8 ml of water. Similarly, we will just go on increasing the quantity of extract. So first tube will have 1 ml, second will have 2 ml, 3 ml, 4 ml, 5 ml and so on, till you reach the last tube which is having 100%, that is full 10 ml extract, and then make up the volume to 10 ml with water. So this is the most dilute solution containing 1 ml extract and 9 ml water, whereas the last test tube is the most concentrated solution containing completely 10 ml extract. Then you shake it for a period of 15 seconds vigorously and then allow it to stand for 15 minutes. You let the foam subside and then you see which test tube has the foam height of 1 centimeter. Now that test tube you will take for foaming index. So foaming index is given by a very simple formula. It is given by a formula 1000 upon A, where A is the test tube, or A is actually the volume which is in the test tube, that is the volume of extract which is there in the test tube having 1 centimeter foam height. So say for example, here in this case my test tube number 3 is having the foam height of 1 centimeter. So my foaming index will be 1000 upon 3. So you know, like 1000 upon 3 is 333.33 will be my foaming index. Now think about it. Say suppose if all my test tubes have foam height more than 1 centimeter, then I say my foaming index is larger than 1000. But even if my last test tube, that is the 10th test tube, doesn't have 1 centimeter, that means the saponin content is very less. I would say the saponin content is, or the foaming index is less than 100. So this is how I measure my foaming index. The last value which we will see today in this session is swelling index. Now swelling index determines the swelling capacity. It is done generally for gums and mucilages which swell in presence of water. So how is it done is, you take about a gram of sample and then as per WHO you take about 25 ml of solvent. Now this solvent is generally water. You kind of shake it and keep it steady and allow it to swell for a period of 4 hours. After 4 hours you see how much it swells. Now there are drugs which will swell more than that. So exceptionally you can even take a cylinder or measuring cylinder which has 100 ml capacity and you can use 100 ml solvent. But as per WHO guidelines, unless otherwise specified, you will take 1 gram of sample in a 25 ml measuring cylinder, pour 25 ml and observe the reading after 4 hours. So say for example isabgol. Isabgol has a swelling index of 35 to 40x. What does it mean? 1 gram will become 35 to 40 ml. So in that case you have to take, you know, like a 100 ml cylinder, because 25 ml won't suffice it. It will kind of swell too much with it. So this is about the swelling index, which is generally done at room temperature. So we saw today WHO guidelines on physical parameters, microscopic, macroscopic as well as organoleptic parameters. So here are a few references you can read through for understanding this, and thank you everyone for your patient listening.